So this is the second video, which summarizes the concepts in the playlist on the reproductive system. The previous video had gone through the maturation of the male reproductive system and its roles. But just a quick introductory comment before starting the female reproductive system in this video is that the focus was on the primary reproductive structures. Those primary reproductive structures are those which are physically involved in reproduction, where gametes are made or gametes are transported. However, there are also what are known as secondary sexual characteristics, traits which vary between males and females, but which don't have a direct role in reproduction. So for example, in humans, the presence of facial hair and the prevalence of hair throughout the body can be a difference between males and females on average, but it isn't directly involved in making gametes. So, facial hair or the depth of the voice, the prominence of the laryngeal projection here, it doesn't facilitate either, sperm and ova fusing with each other. And so during puberty, when there are changes induced by the steroid hormones, estrogen and testosterone, not all are directly involved in reproduction. And so this is not, unique to humans. Obviously, as we consider other animals, there can be courtship songs, which are made by male frogs and not female frogs in this example. There can be differences in the coloration of feathers. There can be a variety of courtship behaviors, etc. And so, as I will revisit in a different set of videos on gender, there are what are referred to as the somatic differences between males and females on average, which are caused by androgen hormones and estrogen hormones, which aren't directly related to reproduction. These are known as secondary sexual characteristics. One quick comment on sweat glands. Sweat doesn't have to have odor, and the watery sweat produced by eccrine or sudoriferous glands would be an example of that. This watery sweat is for cooling. However, at puberty and afterwards, the steroid hormones then cause the activity of another type of gland known as the apocrine sweat glands, which make pheromones. Pheromones are signals that an individual produces in their sweat, which then offers information about themselves and sexual state to other members of their group. And so when dogs meet each other, they smell each other because they are picking up on the pheromones that they produced. Each individual makes pheromones, which communicates whether the individual is male or female. In females, whether they are an estrus or not. In males, whether they are the alpha male who tends to win all of his fights, or the beta or even zeta male, which does not. One can gather information as far as whether the individual is healthy or not, or even pick up on how closely related they are. And so whether it be mice or even humans, we seem to be able to determine innately the degree of relationship we have with an individual based on the aspects of the pheromones in sweat. Now, for mammals in general, many of which are nocturnal, smell is very important, and so the perception of pheromones is important. And there are actually two senses of smell. The one that's in the vomer, its primary response seems the detection of pheromones. So these buffalo, for example, they are picking up on pheromones, even going to the extent of sampling urine to analyze the pheromones which would be present in urine. So for mammals in general, the perception of pheromones seems to be important. However, in the ancestor of the old world monkeys and apes, the sense of smell, which is detected in the vomer, what's known as the vomeronasal organ, it no longer works. And so while most mammals have place a great priority on these pheromones, which are then detected at the vomeronasal organ, in the higher primates, including ourselves, this doesn't work. And so pheromones apparently are detected to some degree at the olfactory epithelium, but their importance is less than what it was. But that would be another example of a secondary sexual characteristic, the production of specific pheromones, which doesn't have a direct correlation with the fusion of gametes and reproduction, but can differ between males and females. Now, as we consider the structures of the female reproductive system, this is not as different from the previous video and the structures of the male reproductive system as one might imagine. And the reason for that is embryos develop reproductive organs before they have decided what gender they are going to be. And so therefore, if one were to consider these two embryos, they start off with the same identical structures, including, because this will be important in a second, this blue Wolffian duct and this pink Mullerian duct. Notice that the gonads in green are in the same sites. It is only then afterwards that these structures, which are then shared between male and female embryos, are then modified to form the structures of the male reproductive system or the female reproductive system. The kidneys rise. The kidneys actually form in the pelvis and have to go up, while the gonads then have to go down. In both males and females, the gonads descend, but in males, they will descend further. Now note that while each of the genders had a blue Wolffian duct and a pink Mullerian duct, the pink Mullerian duct degenerated in males and the blue Wolffian duct degenerated in females. So there are structures which are unique to males and females. So males have a vas deferens. This is the remains of the Wolffian duct. And females no longer have any such structure, even though they once did. And in females, the Mullerian duct forms the fallopian tubes and portions of it fuse to form the uterus. Males have no homologue of this, since that degenerates in male development. There's even an AMH, an anti-Mullerian hormone, which promotes this. So there are unique structures such as this blue and pink tube. But that being said, many of the structures of the male and female reproductive systems are what we call homologous. So the gonads either become ovaries or testes, depending on their genes and the hormones to which they are exposed. Externally, the same tissues either become the clitoris or the penis. These are homologous structures. If a male has problems making or perceiving testosterone, the penis can be so small it could be the size of a clitoris. If a female is exposed to too much androgen, the clitoris could enlarge and be the size of a penis. The same labial folds in females form the scrotal folds in males. And once again, hormonal differences could cause abnormal scenarios where the scrotal folds do not fuse in males and look like labia, or the labial folds begin to fuse in females. And so much of the reproductive structures are actually homologous between males and females. Just as much of the discussion in the male reproductive system focused on gametogenesis, the formation of sperm, obviously then the same would be necessary in females, the formation of ova. This conversation, however, is a bit more involved. The formation of ova in females is far more complex than the formation of sperm. So there are a number of different aspects. So I'll probably then at the end revisit this and summarize, because it will take a while to go through this. Females have oocytes, just as males had spermatocytes. However, the oogonia, which gave rise to these oocytes, they are no longer present. All right, and so in both males and females, there are primordial germ cells which go to the embryonic gonads. Now, these primordial germ cells, interestingly, this is a bit odd, they actually form near the yolk sac, actually outside the embryonic body, and they actually then have to find the gonads. They migrate through the body to find the gonads. If they don't end up in the gonads, then an individual may have, anatomically normal structures, but nevertheless be infertile if these structures do not end up in the embryonic gonads. In both males and females, the embryonic gonads will descend from their original abdominal position. So the primordial germ cells have to move to these gonads. In females, the gonads descend to the pelvic brim. In males, then they have to descend further, going actually outside the pelvic body cavity into the scrotum. These primordial germ cells then produce the spermatogonia, which males retain, and thus, because males retain them, they can then make sperm throughout their entire reproductive lives. The situation is different in females, however. The oogonia begin their residence in the fetal ovary, but then they will then differentiate and become oocytes. And the oocytes will actually then even begin meiosis before a female fetus is actually born. So if one were to consider then the fetal ovary, there are primordial germ cells, which will divide by mitosis, producing the oogonia. All right, they may then form fused masses of cells known as syncytia. And that's what a fused mass of cell is. And so by the point of, say, 2 months of embryonic development, right, the female ovaries then have these oogonia. They will eventually number 6 to 7 million by the 4th to 5th month of fetal development. So just as males have spermatogonia, females have oogonia, maybe 7 million. Now that then is the maximum number of sex cells that they could produce, which is obviously something I'll develop further here. Okay. So 7 million, that's a lot of, say, potential offspring. However, these oogonia then become oocytes. All right, so by the time of birth, there are no longer any oogonia, there are now oocytes, which means that a woman's maximum number of gametes is now fixed. In males, spermatogonia continue to divide, and every time a spermatogonium divides, a spermatogonium remains. Men never run out of the ability to make new sperm, but because all of the oogonia become oocytes prior to birth, there are no stem cells left in the ovary, and thus, as a woman nears menopause, her ovaries are running out of reproductive cells, which can produce sex cells through meiosis. And so, unlike the situation in males, there's now a fixed number of sex cells that a woman could make during her lifetime. Now, it's about 1 million potential sex cells at birth. That's the number of primary oocytes in these primordial follicles. Now that number will be reduced further, but that will come up again later. So the ovaries are where meiosis occurs, just as we call meiosis as spermatogenesis in males, we refer to it as oogenesis in females. There are similarities to what we observed in males. So for example, independent assortment and crossing over occur in prophase 1. Once again, these are both extremely important because as a result, each ovum now has a unique combination of chromosomes and alleles of genes on those chromosomes thanks to these processes. Two individuals have the genetic potential to make more genetically different children than there have ever been human children, thanks to these two processes. So, it greatly increases variation. However, there are some significant differences. Let me introduce those, but these will come up again later. When the primary oocyte divides, the division is not equal. So notice that the cell on the left, the secondary oocyte, is bigger than the cell on the right, which won't even be called an oocyte. It's referred to as a polar body. This one could produce a gamete, this one can't. So while there was a cell division, it wasn't an equal cell division. One daughter cell got most of the nutrients. When then the process of prophase 2, metaphase 2, anaphase 2, telophase 2 occurs to complete the second round of cell division, once again, the division is unequal. So while meiosis in males has one spermatocyte producing 4 potential gametes, in females, one oocyte produces 1 ovum, 1 sex cell, and then 2 or 3 of these other cells, which are referred to as polar bodies. The reason it's 2 or 3 is the first polar body may or may not divide. Now, the video that you're about to see would also be just as relevant to talk about in the next chapter with conception. So let's introduce it here, but then we can, get back to it in the next chapter as well. And so, if you were to observe then meiosis occurring in the ovary, at ovulation, this is the structure which is which is released. Now notice that here we see 2 big relevant cells. We'll get to the aqua ones in a second. So here's this purple cell, that is the secondary oocyte, and then here in yellow, this is the first polar body. So the primary oocyte divided to make 2 daughter cells, but see how unequal they are? This purple secondary oocyte got all of the nutrients. This polar body did not. When they divide the second time, and a few things that just happened, but that I'll have to explain. When they divide the second time, there is 1 large ovum. Here is a second polar body, and this first polar body may or may not divide. So meiosis in females produces 1 sex cell, 1 gamete, and then 2 or 3 of these little cells which will degenerate and not be sex cells, not contribute to fertilization. And so the reason for this is simple. The nutrients here in this cell, all of the resources in the big purple cell, this is all an embryo will have for the first week of its life. And so if one considers the space of time between fertilization, which occurs in the ampulla of the oviduct, and then implantation in the uterine lining, it typically takes about a week in between those processes. It is not until after a placenta starts to form, when the embryo embeds in the uterine lining, that there's new food for the embryo. So this then is all of the nutrients that an embryo will have for the first week of its life. If all of these 4 cells were the same size, that means that this would have been divided in 4, which would give each embryo fewer nutrients and potentially a lesser ability to survive. Now, especially in humans, our typical number of children per pregnancy is 1. And so there would be no reason to divide these resources in 4 if the typical goal was to have only 1 offspring. And so the nutrients are maintained in 1 cell so that the embryo will have the maximum number of offspring, will have the maximum number of nutrients. And so then these polar bodies, they will degenerate and nutrients are not wasted on those. There was one other difference, or I'm sorry, my fault, two other differences. One is, what are these blue cells doing? Well, we'll have to explain that. Those are actually cells of the ovary, which form part of the follicle. Now that's odd because sperm travel by themselves. The male gamete is a solitary structure, but the female gamete develops as part of a much larger structure in the ovary called a follicle. It develops kind of as a team. One other thing is, look at this. This is a secondary oocyte. Now, that's odd. Let me just explain that. That's odd because it's not an egg. It's not an ovum, despite the fact that this is what is released at ovulation, all right? So in males, gametogenesis begins and ends in a short period of time. So, the primary spermatocyte divides, the secondary spermatocytes, they divide again, a tail develops, and we're looking at a little, somewhere between 2 and 3 months, maybe 70 days. That's from start to finish. Now you have the male gamete. This, however, means that even at ovulation, female gametogenesis isn't even over. It actually begins before birth. The primary oocytes actually start meiosis before birth and then pause. And then they wait. Then during childhood, during puberty, then every month more will start again and then pause. And they pause because it is the secondary oocyte which is released at ovulation. This isn't an egg yet. So at ovulation, gametogenesis isn't over. These cells have not finished the last round of meiosis. It is not until the sperm fuses with the ovum that this cell division finishes. These chromosomes then separate, finishing metaphase 2, anaphase 2, and telophase 2, and actually now producing a haploid cell and egg. So the timing of gamete formation in women is much different than in males. It starts before birth and pauses. It picks up during a menstrual cycle at some point in her life, and then pauses prior to ovulation, and it doesn't finish until fertilization. A woman who has never conceived has never truly made an egg, an ovum. She's come close. A secondary oocyte is almost an ovum, but it has not yet finished that last cell division, and the penetration by the sperm is the trigger for that. And so before revisiting this, there are a number of significant differences in female gametogenesis compared to male gametogenesis, such as females no longer have oogony after birth, and so therefore the number of gametes that they can make over the course of their lives is fixed. That when gametogenesis occurs, one cell doesn't make 4 gametes, but rather one cell makes 1 gamete, and then 2 or 3 of these polar bodies which will degenerate. Third, that the ova forms as part of a team. Notice all of these cells here from the follicle, as opposed to the sperm, which travels by itself. And then fourth, the timing is different. So that meiosis will begin before birth and then pause, resume in a menstrual cycle, and then pause, and not complete until the moment of conception. So there are certainly then significant differences in female gametogenesis compared to male gametogenesis. Now, I'd like to just touch on some of those processes in a little more detail, but because it is involved, I will leave the videos in the playlist to explain it a bit further. I just want to kind of hit the highlights of each one. So like I had said, the oocyte, which is undergoing meiosis, it doesn't do so in isolation, but rather epithelial cells of the ovary, they contribute. So these epithelial cells, they then invade these syncytia, and then they form these follicles. So a follicle is then the cell which is undergoing meiosis, the oocyte, but then also these epithelial cells from the ovary which surround it, starting with primordial follicles. The primordial follicles then set the maximum number of gametes that a woman could make during her lifetime. And even when, say, one follicle is undergoing ovulation during a menstrual cycle, the ovary is still full of these primordial follicles. They form during fetal development, and they, in essence, will then wait their turn until a menstrual cycle. And so a primordial follicle has the primary oocyte, waiting the menstrual cycle when it will continue with meiosis. And then there are then the cells of the ovary which surround it, and they touch each other. And these cytoplasmic extensions allow them to communicate with each other. They both will secrete signals which then influence the other. There is a small space called a zona pellucida between them, which forms as well. Now, I and feel a little sheepish a little bit in that as one teaches biology and make videos as I do, one of the things that sometimes happens is that new discoveries occur, which then show that the former understanding wasn't quite as complete as it should be. Now, actually, that's science doing its job. We will never understand nature fully, and then it's only in rooting out, oh, here was a mistake, here we didn't understand this. Well, the female menstrual cycle was certainly not understood as fully as it is now, even not that long ago. And so if you look at anatomy textbooks, even fairly recent ones, there are mistakes, not major ones, but the idea, especially in this video and the next couple, is a little bit different than what was previously appreciated. So prophase 1 can be split into a number of different phases. That's a little bit beyond us here. But one of the phases is where the primordial follicles pause. So meiosis actually starts and ends at the diplotene phase before birth, and that's how the primordial follicles are now paused in the remainder of a woman's life. So these are primordial follicles. Notice how thin the follicular cells are that surround them. And this is kind of the state that they existed in at the end of fetal development. And until a menstrual cycle activates them, they will exist in this phase. Now, every month, about 1,000 of these primordial follicles become primary follicles. You see the primary oocyte will continue meiosis. And notice the difference here in these granulosa cells. They are no longer as thin and flat as they had previously been. And so the primordial follicles will then form primary follicles. First, one or a few granulosa cells will become cuboidal, and then others. When one follicle becomes a primary follicle, it actually inhibits its neighbors, all right? So that, it is not the human condition that all of the follicles mature in 1 month. Otherwise, like, for example, with fish, there would be 1 month where the female, could produce all of her eggs, and then she would be infertile after that. And so one will kind of inhibit its neighbors. And notice during these processes, there's a whole bunch of chemical signals going back and forth. They can pass through the zona pellucida, but these cells are also then touching each other, and they can pass them. So the oocyte can talk to the granulosa cells, and then one follicle can talk to its neighbors, which inhibit its development. And so each month, there are about 1,000 of the primordial follicles, which will then become primary follicles. It was originally erroneously thought that those primary follicles would compete against each other and then mature in a single month. That is now known not to be the case. It can be well over a year before ovulation will ultimately take these. So these earlier stages, it was thought that follicle stimulating hormone directs them in the month that one will be ovulated, but we now know that that is not the case. Primary follicles become secondary follicles, sorry, I thought I had one more, become secondary follicles, which become preantral follicles. And there can be some disagreement in how these terms are used, primary, secondary, preantral. So let's not worry about that, because you could actually, different authors might use them slightly differently. And so as the primary follicle then continues its development, it may take 6 to 8 months, during which time it develops multiple cell layers here, right? And eventually we'll even then develop a space between these follicular cells, and the space is now known as an antrum. And to develop into a primary, to a secondary, to a preantral follicle, to an antral follicle, once again, can take the better part of a year. Also involved in this process is the making of estrogen and progesterone. And so outside this are cells of the ovary, which are helping in making estrogen, and they will be identified as layers of the theca. Now, there's the theca interna, which is making an estrogen precursor. They're here in orange, and the theca externa, which includes collagen and smooth muscle. And it's the dissolving of the collagen and the squeezing of the muscle, which will have a role in ovulation, right? So there's a lot involved, including layers of the theca. And so as a vesicular follicle undergoes ovulation, that outer layer of the theca has a role there. There's blood vessels involved. We'll get back to that. So over months and months, these divisions are occurring until a space develops known as an antrum. And this could be 8 months after the primordial follicle began its development as an antral. So here you have a cell without a follicle without an antrum, but notice this pink region here, that's a space known as an antrum. It's filled with a fluid, a follicular fluid or antral fluid. We could even then, split these granulosa cells into different layers, and the videos do, but I'm not going to cover that. And so the antral follicles, they begin almost microscopic, so they start off at being 1/10 to 2/10 of a millimeter, and once again, this was 8 months, perhaps after the primordial follicles got activated. There are lots of signals going back and forth. So the primordial, the primary oocyte is communicating with the granulosa cells. The granulosa cells are communicating with the oocyte. And then obviously we have other communication. The pituitary is talking to the structure. So there's a hormone called FSH, the follicle stimulating hormone, and luteinizing hormone are talking to the cells of the theca. So there's a great deal of hormonal coordination and regulation of this process. And once again, just to show how complex this is, this video once again just reiterates just how much communication is going on back and forth among all of these various structures. So a lot of hormones are locally active hormones made by a cell to affect its neighbors. And so you can see that the follicle is quite a complex structure with lots of communication going back and forth. It was originally thought that the follicle stimulating hormone was what guided this entire process. It now seems that the maturation of the primordial follicle to the primary, to the secondary, to the preantral, that is largely independent of FSH, that FSH is governing the final stages. So the hypothalamus makes the hormone GnRH, which tells the pituitary to make FSH and LH. These are known as the gonadotropins. Now, they affect both males and females. Follicle stimulating hormone is important for the development of the female follicle, but also for meiosis in males. LH is important in the ovary making progesterone, but also important in the testis making testosterone. So these hormones, while named for their roles in female gametogenesis and the menstrual cycle, are not only functional in females. Males without these hormones become infertile, just as females without these hormones. So follicle stimulating hormone is made by the anterior pituitary under the guidance of the hypothalamus, and there's a number of different things which make it interesting, as far as you can have different forms of it based on which carbohydrates are attached, etc. So I go through the effects of FSH, but we now understand that it's FSH will now guide the latter portions of the follicular development, not the earlier portions. Now, once a follicle reaches a specific point, it is now making FSH receptors, which make it sensitive to FSH. And then there's an interesting FSH window and FSH threshold. So while the follicles are sensitive to FSH, they essentially race against each other to see which ones can get big first. And so once follicles cross this threshold, then their goal is to get enough FSH to develop to the point where they are making their own estrogen. If a follicle doesn't win this race, if it crosses the threshold but is not the one that gets big first, then it will then ultimately degenerate, because once one follicle starts making estrogen, the hypothalamus perceives this, and now the brain will signal the pituitary to make less FSH, and then the FSH levels in the blood falls. The follicles which crossed the threshold, but which did not get big enough to make their own estrogen, now the FSH levels have fallen, and so now they can no longer maintain themselves, and so they will fail. So in a month, a woman typically does not have many follicles undergoing ovulation, but rather one. And that's because they were kind of competing against each other. And the ones which did not get big fastest, they degenerate, and only one of these follicles will undergo ovulation, and then the others will then degenerate. And so out of the 1,000 of primary follicles which formed, maybe a year before, only one will undergo ovulation. Isn't this a waste? Well, just a philosophical point. Humans do two things which, genetically speaking, are stupid. One, they have a small number of children, and two, they live a long time. And genetically speaking, if you put those two things together, what you've essentially said is you will doom your species, because the number of harmful mutations which will accumulate over time will eventually get to the point where your species is just too unhealthy to survive. Mutations occur all the time, and especially while mutations occur in women, most mutations are occurring in men because they undergo so many cell divisions. Each day, a man can make, a few 100 million sex cells. Now, every single cell division gives a chance for mutations to accumulate. And if each day men are making hundreds of millions of sex cells, by the end of a man's life, his stem cells have accumulated mutations. And so, therefore, the number of mutations would increase. And now, the same is true of women, but to a lesser degree, because the number of cell divisions is less. Each of us, as far as we can tell, had over 100 mutations that we inherited from our parents at the moment of conception that were not present in the body cells of our parents. So mutations happen all all the time. Now, mutations are often good. I mean, I'm not a regular old ape. I'm a variant ape. So at one point, mutations in our ancestors modified us. Mammals are mutant reptiles. Reptiles are mutant amphibians. So I'm not knocking mutation. This is what allows change over time. But many mutations are harmful. The way that our species survives is that we ask sex cells to compete against each other. So in an ejaculate, there are tens of millions of sperm, hundreds of millions of sperm, and they're competing against each other, swimming an incredible distance, the length of the vagina, the uterus, and a fallopian tube. Sperm which have major mutations are much less likely to be the first ones to reach the secondary oocyte. So by asking sperm cells to swim such an incredible distance and compete against each other so that only one will win this race, a species is essentially trying to eliminate less healthy sperm and maximize the likelihood that the most fit sperm will fuse with the oocyte. But in a less visible way, the same is true of the oocytes, because in fetal development, there was, for say, the 7 million oogonia, this resulted in, say, maybe 1 million oocytes. From the 1,000 which will start development in a given month, only 1 will finish. And so just like there seems to be a selection against male gametes so that only the best are selected, the same seems to be true in female gametes. And so therefore that seems to be the way that we ensure that even though we live long and thus accumulate lots of mutations, especially in males, and that we have a small number of offspring, nevertheless, these represent the best of our genes. Just an interesting point, the female follicle makes 2 important steroid hormones, estrogen and progesterone. The interesting thing is there are 2 signals from the pituitary gland. FSH promotes the production of estrogen. LH, the luteinizing hormone, promotes the formation of progesterone. And there are actually 2 different cells which produce each of these hormones. So estrogen and progesterone are made from cholesterol over a long series of steps. So for example, what you just witnessed is a cell will make testosterone, and then from testosterone, the last enzyme converts testosterone into estrogen. But these are these chemical processes are located in 2 different cells. So here you see the theca interna cells here in orange. They convert cholesterol into things like testosterone, whereas then the testosterone then migrates to the mural granulosa cells here in aqua that then receive FSH and convert androgens into estrogens. And so the making of estrogen actually requires both FSH and LH, 2 different gonadal hormones, gonadotropin hormones from the pituitary, and occur then in 2 different cells. The first half occurs here in these theca internal cells, and their precursors then go to the mural granulosa cells to be completed. So once again, there's a great deal of complexity here. That last thing was, if that last step isn't done, where testosterone is converted into estrogen, that can, because of a mutation, that can actually have a masculinizing effect, because now a woman isn't making much estrogen, but is making a lot of testosterone instead. The hormone LH is made by the anterior pituitary gland, and it spikes in the bloodstream midway through the menstrual cycle. And its spike then, this is the signal for ovulation. So ovulation is triggered by a great increase in the luteinizing hormone made by the anterior pituitary cells. This follicle will then start to dissolve the collagen, all right? So the collagen starts to dissolve in the theca externa, and that makes the wall of the follicle weaker. And then these smooth muscle cells will be stimulated to contract. And then by contracting on this follicle with this fluid, which now begins to develop pressure, the pressure will then cause the bursting of this follicle, releasing the secondary oocyte and some cells which surround it, which then begin to travel through the oviduct. Right? So that is the process of ovulation, and it is triggered by triggered by that surge in LH. Some of the cells of the follicle, of those granulosa cells, they continue to surround the oocyte, and those were the aqua cells that you saw at the moment of fertilization. But some of the cells remain. So notice all of these aqua cells of the follicle, they are not released at the moment of ovulation. And instead they become modified to form the corpus luteum, the yellow body. The yellow body then is an endocrine structure which makes the hormone progesterone. Progesterone is what will maintain the uterine lining by causing cell divisions. So it's under the influence of the corpus luteum in the second half of the menstrual cycle that the functional layer of the endometrium reaches its thickest point. So here's the corpus luteum. It has a bit of a yellowish color, hence its name, corpus luteum, meaning the yellow body. So the corpus luteum then secretes the progesterone, which makes the uterine lining increase its thickness, because if conception occurs, then the embryo needs to implant into the uterine lining, and so progesterone helps to prepare the uterine lining for possible conception. If conception doesn't occur, then the corpus luteum will become the white body, the corpus albicans, and then just degenerate from there. And so I had mentioned that there was some misunderstandings and some growth in our knowledge with the menstrual cycle. One of the keys was that it is not FSH which governs the entire process of follicular generation. The first 6 to 8 months occurs largely independent of FSH. And because it takes place in so long, it's not all occurring in a 1 month period. And so thus then the ovaries would have follicles at many different stages of development. So that wasn't fully appreciated. So the corpus luteum secretes progesterone, as I had mentioned. And if one, we'll get back to this. All right, so we'll get back to that in a second. Once ovulation occurs, the secondary oocyte, remember it is not an ovum yet, is released then into the fallopian tube. Now the fallopian tube has an expanded region, the infundibulum, whose finger-like projections, fimbriae, try to catch the oocyte, because if the oviduct does not catch the oocyte, then the oocyte could be floating around in a woman's pelvic body cavity. And if it were fertilized, this could cause an ectopic pregnancy, which could be life-threatening. So the expanded outer edge of the oviduct, wants to catch the oocyte after ovulation. Once it does, then the oocyte will pass along the length of the oviduct from the infundibulum to the ampulla to the isthmus, and it moves in the right direction, in part because of the fimbriae, which help to guide it, but also peristalsis occurs over the length of the oviduct. And then also the epithelia of the oviduct are ciliated. And so the motion of these cilia creates a current which helps to guide the secondary oocyte towards the uterus. It narrows, the isthmus of the oviduct narrows, and sometimes this narrowness can then be a problem where a woman might require a surgery because the isthmus of the oviduct is so narrow that it doesn't allow secondary oocytes to reach the uterus, and therefore is a cause of infertility. So there was an enormous number of changes occurring in the ovary over the course of a menstrual cycle. But then also there are changes in the uterus as well. So just a quick introduction. The uterus has layers. There is an outer serous layer, the perimetrium. This allows the uterus to move relative to neighboring structures without disturbing them. The thickest layer is the smooth muscle of the myometrium. The contraction of this smooth muscle is involuntary, and this is what will expel the infant during childbirth. Its contractions also help to expel menstrual flow, and then even after intercourse, contractions induced by hormones in semen help to create contractions which help to propel sperm. So that's the myometrium. The innermost layer of the uterus is the endometrium, which then has two sublayers: a basal layer of the endometrium, that's depicted here in gray, and then a functional layer of the endometrium in magenta. The functional layer is what will develop over the course of a menstrual cycle and then be shed during menstruation. So here you see the functional layer of the endometrium being shed during the menstrual phase, and then estrogen will promote the development of a new functional layer of the endometrium. So this is menstruation. You can see the ragged edges here as these cells are breaking down and being shed. But it is estrogen, which is developing from that dominant vesicular follicle, which causes then the proliferative phase, where the cells of the endometrium undergo a great deal of cell division, and the functional layer of the endometrium becomes much thicker. That will last, this proliferative phase and the development of this functional layer, until the moment of ovulation. After ovulation, the cells which remain in the ovary, that becomes the corpus luteum. The corpus luteum then secretes progesterone, and then progesterone causes the secretory phase of the uterine lining. There's more cell division. In fact, this is when the functional layer of the endometrium gets to be its thickest under the influence of progesterone, but also then there is the secretion from uterine glands, which will help nourish the embryo. So here you can see the spaces, the ducts of uterine glands through which a number of secretions can reach the lumen of the uterus. And so during the menstrual phase, it is the functional layer here in magenta which is shed. The basal layer will be maintained, and then there will be a pre-ovulatory phase or proliferative phase. This basal layer was the only layer of the endometrium present for a bit, but then under the influence of estrogen, a new magenta layer will develop, this functional layer, the stratum functionalis, and then under the influence of progesterone from the corpus luteum, this reaches its thickest point in the second half of the menstrual cycle. It is so thick because after ovulation, a week later, a fertilized embryo might be seeking implantation. And so then that thick layer of the functional endometrium would be the ideal site for implantation. If fertilization does not occur, this corpus luteum becomes the corpus albicans. Blood vessels at the base of the stratum functionalis constrict. These cells then die, and they will then be sloughed off as the next month's menstrual flow. And so obviously this is very complex because the menstrual cycle not only involves changes which occur in the ovary as the follicles develop, but also there are changes in pituitary hormones, FSH being made in the first half of a menstrual cycle, a peak in LH at the midpoint of the menstrual cycle. But then there are also changes in the uterus as well, as the functional layer of the endometrium from the previous month is shed, and then under the influence of estrogen and then progesterone, a new functional layer of the endometrium were to develop. So that's the synopsis of the videos in this playlist. I have other playlists which go through other topics such as breast cancer, which is obviously related, but once again, I cover elsewhere. And so these videos offer more details than what I had gone through here. So feel free to obviously visit individual videos in this playlist if there was anything that you wish more information on.